I lost the sharing. I don't know if that's an issue. I tried to share it again. I hope you can see it. So, as I said, the first reaction is the coordination of the isocyanide. The second reaction is the conversion of the azide through the reaction with the phosphine to the amino group that then attack nucleophilically the isocyanide carbon and form the final carbene complex. And here you can see also X-ray structure of the complex. Then we said, well, of course, isocyanide are very interesting for the possibility to convert them into different ligands, like, as we said, carbenes. But what if we use the isocyanide ligand simply as a ligand to stabilize the metal center? Well, of course, that's possible, but we also know that to stabilize the metal center, the chelate effect is important. So we decided to investigate the possibility to use our chemistry to prepare chelating the isocyanide. And so what we did was to follow a very old synthetic procedure, which was proposed by Angelici, which is a very important scientist in this area. But as you can see, the series of reactions that are necessary to get to the chelating the isocyanide is very long, and the final yield is very poor. But quite recently, we came up with another possibility, synthetically speaking, which is the reaction which go through the opening of an oxazole by reaction with a strong base like butyl lithium. Okay, when you react the oxazole with butyl lithium, you deprotonate the oxazole in the carbon between nitrogen and oxygen, and these generate an equilibrium between the cyclic form and the open form, which is also an isocyanide form. And then we can use the oxygen, lithiated oxygen, to link this fragment to a bridge. So what we did was to follow something that already in 2010 Oliver Reiser did, applying the chelating ligands that he obtained to iron and to palladium, but to our platinum. So we started by synthesizing the chelating the isocyanide that you can see at the top. The reaction is one pot, but the yield, since we are using very reactive species, is relatively low. But as you can see by the number of possible bridges between the two isocyanide units, which are represented here by the letter Y, the possibility to synthesize an entire library of ligands is huge. So we prepared all those ligands and we prepared the corresponding platinum dichloro complexes. Here you can see in the picture on the right one of the possible structures that we obtained. And then we thought, why not to try the stability of these complexes and their activity also in catalytic reactions. And so we decided to study the catalytic reaction, in particular the Fujiwara reaction, which is the hydroxylation of alkenes. So the activation, basically, of the alkene toward the hydroxylation. And what we observed is that our complexes, either in the chloride form or in the methyl form, they are quite active, reaching yields which are around 90%, but they're also quite selective, because in brackets you can see the alkene conversion that tells you the amount of monoarylated derivative compared to the possible byproducts of the reaction. And you can see they are very, very high, even compared with other platinum complexes or species, which can reach similar conversions, but definitely lower selectivities. And so then we extended the catalytic activity of these species, studying different aryl groups, different alkenes, and we obtained the results that you can see. And in some cases, as I said, the yield reaction is quite high. We're still not comparable to some palladium complexes and some carbene palladium complexes, but still we can prove that these complexes are stable even in the harsh condition of this reaction, and so we can obtain the desired product with good selectivities. The second part of my talk is dedicated to a more recent work that I performed, and which was started early in my career during my PhD, but then followed. And for us, it's very important because it's based on a particular catalytic activity of palladium complexes. We studied since very, very long ago the possibility to use platinum as a catalyst for oxidation reactions. Well, why oxidation reactions? Well, we know that oxidation reactions are very important generally in the preparation of organic derivatives and preparation in the modification of some moieties in organic compounds for further modifications. But one of the problems with oxidation, in particular organic oxidation, is usually the need of very strong oxidant, imagine like peroxides, organic peroxides and so on. While there is a need to move toward more benign, more green oxidants, we can imagine, well, oxygen would be the perfect one, but also hydrogen peroxide is a very good candidate because, well, it's a commodity that we can obtain easily, and by oxidation, it gives only water as byproduct, so a very green byproduct. So we decided to study the possibility to use hydrogen peroxide in organic oxidation. But the problem with hydrogen peroxide is that it's a quite, let's say, bad oxidant. It needs activation, or at least we need to activate the substrate to be able to use it as oxidant. So why did we decide to use platinum? Well, because platinum, we know, gives usually quite stable complexes. It's relatively inert as a metal. And particularly so if we stabilize it with a chelating ligand like a diphosphene. And then we know that usually it's compared to, for instance, to palladium, it doesn't give easily oxidative addition or beta hydride elimination. And, which is very important compared to other metals like iron, copper, and so on, it doesn't give one electrode redox couples. Okay, so usually its chemistry is related to two electrons exchange, and this is very important because it allows to avoid the decomposition of hydrogen peroxide. So what we studied was a series of complexes with the structure that you can see there. They are platinum-2 complexes, so we expect the typical D8 square planar geometry, but with a chelating diphosphene ligand, which is something that gives stability to the complex, but also allows to vary, is steric, so the space occupation, but also electronic properties on the metal center, as we'll see very soon. But also leaving one free site, the one you can see in green, indicated with a Y letter, but using the fourth position for an ancillary ligand with electron withdrawing properties. So a species, usually a ligand which is based on carbon, which can withdraw electron density from the metal and increase its Lewis acidity. The study started far before I joined the research group, and in particular in the study of epoxidation. It was observed that with simple diphosphene ligands, we can imagine like DPPE, so basically is a 1,2 the diphenyl diphosphene, and with the trifluoromethyl group coordinated to the platinum, the complex was active toward the oxidation of alkenes, and so the epoxidation of alkenes, with a mechanism that was discovered was basically a double mechanism. In fact, you can see on the left that platinum acts as an activant toward the alkene because it coordinates the alkene and so make the alkene more prone to oxidation, to attack from the oxygen of the peroxide. But on the cycle on the right, you can see that it also acts as an activator for hydrogen peroxide. And in fact, coordinating the hydrogen peroxide, it activates toward the attack on the alkene and for the formation of the epoxide. So starting from this observation, we decided to explore a bit more this kind of chemistry because it was very interesting. And so we thought, well, what happens if we create a species which is already, which has already two coordination position free? And so we started from the dichlorophosphine complex by abstraction of the 2-chloride with a silver salt. And depending on the type of silver salt that we used, if it was tetrafluoroborate or triflate, we could obtain two different types of complexes. The first type was a dimeric with hydroxyl bridge ligands complex. The second was a simple monomeric bistriflate ligand complex. But we also decided to change the nature of the fluorinated ligand to see if we could change the electronic and steric properties on the metal center, and so see if it was possible to change the activation of the molecular species involved in the oxidation reactions. And so we studied, instead of the trifluoromethyl, the pentafluorophenyl groups. So it is possible to synthesize the pentafluoro derivative very easily. You can see at the bottom that by starting from a dimeric pentafluorophenyl starting complex, by reaction with a diphosphene, we can obtain the chloro complex, which is a central one, and then by extraction of the chloride, in this case, the type of silver salt wasn't really important. We can free the fourth position, which is the free coordination site for catalysis. And so we started with the hydroxyl bridge ligands, and we observed that actually they were active not only in epoxidation, but mainly in the Baeyer-Villiger oxidation of ketones. We all know the Baeyer-Villiger oxidation of ketones is a reaction that allows the conversion of a ketone to the corresponding lactone, or in generally speaking, of the corresponding esters, and then it can be very useful, for instance, to prepare precursors for polymers and so on. So what we studied was a series of complexes with a simple diphosphene, phenyl diphosphene, with the structure that we can see on the left. But we also wanted to see if there was an effect of the electronic properties of the metal center through the phosphine. So what we did was to synthesize a series of DPPE derivatives of diphosphenes, bearing on the phenyl groups a different number of fluorine atoms. We know that fluorine in the periodic tables is the most electron withdrawing the most electronegative element. And so we expected the increasing the number of fluorine on the phenyl groups bound to the phosphorus, we could also change the electronic properties of the metal. So how did we then tested that this hypothesis was correct? Well, both electrochemically, we performed cyclic voltammetry, and spectroscopically by using a probe coordinated to the metal center. If we see at the cyclovoltammetry at the bottom left, we can see that there is a constant effect of the increasing number of fluorine on the reduction potential of the complex. Here we're talking about the cyclovoltammetry of the complex, okay? This means that the groups bound to phosphorus, to the phosphorus of the phosphines, have a strong effect on the general electronic properties of the metal center. On the other hand, on the right, you can see what's the effect also by simple infrared spectroscopy. What we use here is again an isocyanide, but this time the isocyanide was used as a probe. The isocyanide bond, in particular, has a very specific band, okay, which is very sensitive to the electronic properties of the metal center. So if we start from the same isocyanide and we change the properties of the metal center, what we can see is the variation in the position of the infrared band of the isocyanide, which by coordination is the triple bond between carbon and nitrogen is basically strengthened, even if we don't expect this from the coordination. And the size of the shift of the band, we can imagine it's correlated to the electron withdrawing properties, the Lewis acidity of the metal center. And here we can see from the table we have exactly the same effect observed by cyclic voltammetry. Okay, so an increase in the shift by increasing the number of fluorine on the ligand. So we decided also to study the effect of the steric hindrance of the ligand on the properties of the compound. And we see here, by comparing different alkyl groups allows you, of course, to increase very easily the bulkiness of the phosphine, moving from simple methyl groups to a very bulky terbutyl or cyclohexyl groups. And what we observe here was something quite interesting, that you have a sort of volcano effect. So it's not the most hindered ligand that gives the best results, but it's the ligand which has steric hindrance which is like average. This means that it's a ligand that allows to produce the active species. We start from a compound which is a complex, the catalyst, which is really a pre-catalyst because it's as you can see, the hydroxyl bridge is dimeric complex that in solution during the catalysis has to split, okay, to generate the free site for coordination of the ketones. In this case, if we have a diphosphene which is bulky, this allows for an easier break of the dimer and so an easier formation of the active species. But if we increase too much the hindrance of the diphosphene, what we end up is to hinder the access to the metal center of the substrate. And so we start to reduce the effect, the catalytic effect of the complex. So basically what we observed was that the isopropyl derivative was the one which gave the best results. Okay, even if in this case the different phosphines had quite similar electronic properties. So we can conclude that for this particular type of dimeric complexes, we have a double effect. One is electronic. As much Lewis acidic is the metal center, the most activities. The other is the effect of steric hindrance. So we need some steric hindrance, of course, to form the active species, but we don't have to hinder the access to the coordination site. Here you can see the proposed mechanism. So as we said, the first step is the splitting of the dimer, the formation of the active species, which can coordinate both the ketones and the hydrogen peroxide, and then through a concerted mechanism form the final product, which is the lactin. But as we said, we also studied the pentafluorophenyl derivatives, which is similar to the ones that we saw prepared in the past, but with a group which is definitely more bulky. Okay, so in our mind, it was a way to, as we said, vary the access to the metal center of the substrate. And what we observe here is that using this particular ligand, we could selectively form epoxide in terminal position. We all know that in the epoxidation reaction, the most substituted alkenes are more reactive, okay? But because they are more electron rich, of course, but this means that terminal alkenes are usually the most difficult to be converted. And what we observe with our complexes is that we could convert selectively and with high yields only terminal alkenes. Okay, we studied a whole series of different diphosphenes, and we came up with a proposed mechanism, which is the one you can see there, in which the pentafluorophenyl has a double effect, which is the one of regulating the access to the metal center by the alkene, okay? And being bulkier than simple difluoromethyl, it can allow the access only of terminal, less hindered alkenes. On the other end, through the fluorine, it can form hydrogen bonding to the hydrogen peroxide, and so by proximity, it makes it easier for the peroxide to attack the coordinated alkene. And so in this way, forming the epoxide, which is the product of our reaction. Which is, as I said, the reaction is very selective, radioselective. If you compare it with a quite common oxidant, which is the metachloroperbenzoic acid, we can see that the organic peroxide is selective only or basically toward the internal alkene, so double bonds. While with our complex in hydrogen peroxide, we obtain 100% radioselectivity toward terminal. Okay, and this means that this possibility gives the possibility to create species which are very difficult to be produced through other and more common methods. We studied, of course, the diastereoselectivity, but more importantly, we studied the possibility through the use of a chelating chiral diphosphene to produce also enantioselectively epoxides. You can see here some of the results. The enantio enrichment is, let's say, modest for some of the phosphine studied, but in some cases we got very, very good results. In particular, with S-Kiraphos, which is the first one that you can see on the left, we reached enantioselectivities and EEs up to 98%. But what we thought then, and here you can see some of the results that we obtained, also in alkene isomerization, which is another reaction. I don't want to dedicate too much time to these species, but this is just to say that these particular complexes, so the pentafluorophenyl complexes, are active in the activation of multiple bonds, not just in the oxidation. So By varying the type of diffosphene, we can also have a reaction like alkene isomerization, which are important in some other aspect of organic synthesis, like the preparation, of course, of perfumes and so on. But what is more important was to eliminate one of the issues, the green issues of the catalytic reaction that we considered earlier. All the reactions that I showed earlier were performed in the presence, of course, of hydrogen peroxide, but using an organic solvent. And we know that organic solvents, in particular chlorinated solvents, are very harmful, and so they have to be avoided in terms of environmental impact of our reaction. And so we thought, with the help of our colleagues in the Venice University, to study the possibility to avoid, to limit the use of organic solvent by using micelles. Okay, we know that when we add to water a surfactant, it tends to form micelles in which the apolar chains of the surfactants tend to interact together, showing to the solution only the polar heads. Okay, so we thought, what if we use only water as a solvent, but allow for the presence of our complex, which is relatively less polar. It tends to dissolve easily only in organic solvents, but also the substrate, which are usually apolar molecules, to interact it with the oxidant, but within the enclosed system formed by a micelle. Well, we can get many good effects by using this system, because the micelle can be imagined like a small reactor within which we can perform the oxidation reaction. Okay, so it can enhance the catalytic activity, definitely can enhance the solubility of the reactants, but it can also create a more concentrated system within the micelle. So if we increase the concentration locally of both the catalyst and the substrate, we can also increase the on one side the speed of the reaction, the rate of the reaction, and on the other the selectivity. So what we did was to study the effect of the interaction of the complexes that we prepared. Here you can see the three types of complexes that we studied with a simple surfactant. We started with simple SDS, so sodium dodecyl sulfate, and we studied before considering the catalytic activity, what was the position in the micelle that the complex could take. And this is possible by using like gradient NMR techniques. And what we observed is that the highest was the charge on the complex, and so on the metal center. The closest the complex was to the polar head of the surfactant, as you expected. While the more apolar the complex was, so for instance, the pentafluorophenol complexes, which are quite apolar or less polar than the others, tend to reside within the micelle, interacting with the apolar side chains. And so we started studying also the catalytic activity, and if there was any improvement in the catalytic activity and selectivity by using these systems. We studied both the epoxidation and the Baeyer-Villiger oxidation reactions, and what we observed was, on one side, an increase in the yield, even usually quite mild, but definitely a great increase in selectivity, in particular in enantioselectivity, okay, as you can see from the tables. So why do we expect more active catalysts in the micelle? Why is the selectivity increase in the micelle? Well, on one side, we have that concentration effect that we said. Moving from the simple organic homogeneous solution, where you usually use a substrate in the less than molar concentration and the usually millimolar concentration of catalyst, we can increase the concentration of both within the micelles of at least one order of magnitude. Okay, and these of course have an effect both on rate and yield. But we have also to consider the fact that the micelle is a very peculiar system. And in fact, we can imagine the micelle as a microreactor in which the surface of the micelle is more polar, the inside of micelles is apolar or less polar. And so we have the effect of the position of both the substrate and the catalyst, okay, and the interaction with the different polarity section of the reaction system, which is the micelle. And so what we observed is, of course, that the enhanced selectivity and the anti-selectivity of the complexes within the micelle was due to a modification in the energy profile of the reaction. And so we have a different Gibbs energy of reactions moving from the simple homogeneous solution to the micelle reactor. Okay, so within the micelle, we can both use the gradient in polarity between the external part and the internal part, but also, as we said, the fact that it's a confined space. And if we put inside this very small and confined space a species which is already chiral, okay, and so not isotropic, we can create in that space for the substrate an anisotropic and so enantioselective space. Finally, we started, since we moved from organic solvent toward water, we decided to study also hydration, simple hydration reactions. So we don't need oxidant, we don't need other specific reagents, but only water. And we studied the hydration of alkenes. In this case, we increase the number and selection of possible ligands, and we obtain very, very good results. But let me quickly, because time is passing very quickly, move to the last bit, which is very important in our research and is also something that we are still studying, which is the application in bioinorganic chemistry. We all know, I won't be long in this, that platinum is the, let's say, the king metal in anti-cancer. Sorry? In anti-cancer research. We know that during the decades, many different platinum species have been developed as drugs against cancer. Cancer is one of the main, reason for death around the world. But since 78, earlier in the 60s when it was discovered, and then 78 when it was approved for use by FDA, cisplatin has been and still is one of the main actors in the fight against cancer. Cisplatin is a very, very simple molecule, but is a very, very effective, at least toward specific types of cancer. As I said, cisplatin is basically considered the first generation of platinum drugs, and many others have been developed in the years. In the second generation, you can see the simple chloride, which are the easily exchangeable and so leaving groups on the metal is exchanged for a diacid group. But in the third generation, even the simple ammonia molecules have been substituted for more stable chelating diamines. in the third generation. So moving from the first to the second to the third generation, we usually have a limitation in some aspect, which is, for instance, the liability of the living groups, which is lower, of course, for chelating oxygenated ligands compared to simple chlorides. But we also have usually a wider spectrum of activity or lower side effects of the complexes. What's the activity of cisplatin? We all know the main action is due to the coordination to the N7 position on a guanosine residue along the strand of DNA, and these provide modification in the structure of DNA, but also, of course, the disruption of the transcription and in the inhibition of transcription, which then leads to apoptosis and cell death. So what we considered is that in the big panoramas of different possible platinum complexes, there are some that are quite interesting. As you saw before, most of the approved drugs have a cis square planar geometry, but in the literature we found that even some trans platinum complexes can be active. And so we thought, with all our knowledge on platinum chemistry, to consider the possibility to work on nitrile compounds, both cis and trans nitrile platinum compounds, which can be converted in many different derivatives by nucleophilic addition on the carbon of the nitrile ligand into a series of different ligands, which are like iminoether, amidine, and iminothioethers. And so I want to show you very quickly the results that we obtain with such complexes as anti-tumor compounds. And so the reaction, as I said, is relatively simple, is the activation of the nitrile, usually benzyl nitrile or benzylic nitrile toward by coordination to platinum, and then the nucleophilic attack of a nucleophile, which can be an alcohol, an amine, or a thiol, on the carbon to form the, in this case, the corresponding iminoethers or the, as I said, amidine or iminothioethers. As you can see, the attack can happen both in a Z or E configuration. They tend to form, and their stability during time, and so their inertness depends on the type of nitrile that we use to start with, and on the group that we attach through nucleophilic addition. Here you can see a couple of examples, but most importantly, what we observed that in the case of iminoethers, the cis geometry is still important. And in fact, the cis derivative that you can see here, obtained by reaction of methanol toward the benzyl nitrile, is very active toward a panel of different cells, and most importantly, it shows IC50 measured by the MTT test, which are far lower than the corresponding ones of cisplatin. So it's a good, definitely a very good alternative for these type of cancers compared to cisplatin. But of course, we have also to test it toward cell lines which were inactive, resistant to cisplatin, and to our joy, we observed that the iminoether derivative that we obtained as ERA, so a cross-resistant profile, which are far better than other complexes, and in fact, it can overcome cisplatin resistance. But so then we move toward other derivatives. We, by reaction with different amines, we can obtain the amidine derivatives. Depending on the type of amine that we use, whether it's a primary amine or a secondary amine, we can obtain more easily the Z derivative or the E derivative. Usually the Z derivative can form more easily if some kind of interaction like hydrogen bonding can form. The E derivative usually is more thermodynamically stable because it allows for the more bulky groups to be far away from the metal center. But also in this case, we obtained that for at least one of the derivatives, we got some very good results. And in this case, and which is very interesting to observe, with amidines, the geometry of the metal center is completely different. We have the most active species, which are the trans derivatives. Also in this case, in a huge panel of different cells, we obtain very good results, comparable or most of the time better than cisplatin itself. And even in this case, we obtain a good cross-resistance profile. So the compound was active, definitely more active toward resistant cells than cisplatin. And so it could represent, again, a good alternative to cisplatin. Finally, we studied the reaction with the thiols. Thiols behave quite similarly to alcohols that we saw before. So we obtained the iminothioeters. Here you can see a couple of examples. In this case, basically, most of the complexes we obtained were in E configuration. And again, we observed that for this particular type of ligands, the trans geometry proved to be the most active. And again, with an activity which is much higher than cisplatin itself. Of course, even in this case, we studied the cross-resistance towards cisplatin, observing a very good activity of the complex, which is basically the same for resistant cells as for non-resistant ones. Okay, so basically we can summarize saying that the complexes that we prepared, which are in terms of structure of the ligand quite similar because they are, have all been prepared from nitriles, have some features which are similar. Not the geometry on the platinum, because we saw that the iminothioeters are more active in cis configuration, while the amidine and iminothioeters in the trans. But if you consider the ligand itself, we can see that all bear a phenyl group, okay, which impart lipophilicity to the complex, and this can be important in the transport of the or interdiffusion of the complex through the cell membrane, which is lipophilic. But also we see that they are all in E configuration, which we saw is the more stable one. So we can expect that the configuration is maintained also within the cell. So to just to give an idea of what we are now doing, but we are also moving toward different metals than platinum. We are also considering other phosphines and phosphinic ligands, studying the PTA base, so this phosphinoadamantane ligand, which is water soluble and which can be easily modified by alkylation on the nitrogens. And we are currently studying these systems, which allow the coordination of platinum only through one side, but already gave some interesting results on the treatment of osteosarcoma in dogs, and which is now being studied more thoroughly. So generally, we can conclude that platinum is definitely a very expensive metal, but it can be an actor in many different, let's say, fields. As we see, we can study it in terms of activation of ligands, like we did with isocyanide to produce carbene ligands. But it has also, still believe a very important role in catalysis, not just as a metal platinum, like in platinum carbon catalyst or electrocatalyst, but also as a complex compared to palladium. Catalytic activity is more limited. Definitely, palladium we know can give many more different reactions, but it's very selective. Okay. For instance, I'd like to remind you, in the epoxidation of terminal alkenes is 100% selective, so it's a very, very good catalyst for that specific application. But of course, the biological activity is also a thing that we have to consider, because platinum is still the most important metal in anti-cancer drugs research, but we can't forget that there are many other metals which are now being studied. Platinum, of course, is being studied in the 2 oxidation state, but also 4. But gold is becoming more and more important. We know that gold is already used in medicine as, for instance, anti-arthritic drug, but it can be used there are many studies nowadays on its use as an anti-cancer compound, and of course copper. Copper is a metal in the first transition row, is much cheaper than the other, and it's proving very, very interesting nowadays in treatment of cancer. And actually, some of the research that we started collaborating with India and with the Dr. Ramachandran was on copper, and we are still studying copper as the metal nowadays for these applications. So I would like to end my presentation by, of course, remembering very nice times in the past when we had Indian guests, not just Dr. Ramachandran, of course, but also the great Professor Natarajan. And I would like to thank also all the other friends that we still have in India. I would like to thank again the organizer. I would like to thank all of you for being here today, and I renew my welcome if any one of you would like to pay a visit to Padova and so try one of our spritz, okay, which is the most important drink in it, part of it. So thank you again, and please, if you have any question, I'm here. Even in the future, I can send the presentation to Dr. Ramachandran. Can share it with you. Here in the presentation, you can see the QR code for our department website, where you can see all the different areas of work, which are in all aspects, basically, of engineering. And of course, you can contact me through the email. Okay, thank you again. Thank you. I'm sure that Dr. Ramachandran and the organizer can then, so that they did share on the chat the assessment form with some questions. Most of them are related to the aspect of platinum chemistry that we have discussed today. In the presentation that I'm going to share, there are some, as you probably saw, there are some references to reviews and articles. I would suggest for who's interested to go and read them.
Good evening, sir.
So, if I can answer to Dinesh Kumar, as if we synthesize all the complexes in the research that we reported, yes, they have all been synthesized in our laboratories. Yes. Cheminformatics softwares. I must admit that in the past our laboratory was mainly a synthetic laboratory, and for characterization, so spectroscopic characterization. So we used a lot NMR, of course, infrared, as I showed in my presentation. But through collaborations, for instance, with Professor Pombeiro in Lisbon, we started also the electrochemical characterization, which nowadays is quite important. If we imagine, for instance, the platinum-4 complexes, the octahedral platinum-4 complexes as anti-cancer, they are just the prodrugs, so they become active only when entering the cell and being reduced. So the electrochemical properties are paramount in this application. But more recently, I would say in the last 5 years, we started collaborating with other groups here in Padova for the application of computational method to study the stability, but also to predict the, for instance, the spectra of some of our complexes. Yes. How long it takes to prepare the complexes? Well, it really depends on the complexes. So most of them less than 1 day. Most of them are very, very easy to prepare. For instance, the diphosphine complexes can be prepared in half an hour, 1 hour. Okay. Reactions are usually quite clean, let's say, so the number of byproducts is very limited, and so also the workup is very limited, luckily. For some of the anti-cancer complexes that I showed earlier, where you have different possible geometries of the product, the E and Z geometries, you need chromatography usually to separate, because there is always a formation of both. Okay, one is major if you work for, if it's the kinetic product, one is major if you leave more time, so it becomes the thermodynamically more stable product, but then you usually have to separate them by chromatography. What are the evidences needed for proposing this kind of catalytic mechanism, and what are the methods used, like trapping intermediate? Well, sometimes we could visualize. They are relatively slow reaction, let's say. They take, let's say, hours. So NMR can become a technique that allows you to evidence the formation of some definite species in the solution. Okay, and in some other cases, as I said, this is more recent. We based our mechanism also on computational methods. So usually what we see, what we did in the past was to perform catalytic tests by changing, basically to analyze the effect of the concentration of the oxidant, the concentration of the substrate, the amount of complex. So basically only through catalytic tests and determination of the starting speed or rate of reaction. More recently, we're moving more toward the computational side and the support of calculus. Yes. What are the other methods that could activate isocyanide? Well, usually isocyanide can be activated by basically any transition metal. We have to remember that isocyanide are relatively hard ligand. They are carbon-based, so we can obtain complexes basically of every metal in the transition series. In fact, we prepare some complexes, for instance, of tungsten, of chromium, of copper. So what's important is that the carbon of the isocyanide interact with the Lewis acid. Even if you react it with proton, basically, you can activate it toward nucleophilic attack. Okay. What's the advantage in using transition metal? Well, if you're interested in preparing metal complexes, in that case, you already have the coordination of the ligand through the isocyanide, and then you form the final ligand, which is for instance, the carbene, straight on the metal center. So we use both the activation and the previous coordination to the metal center. How long did it take to obtain crystals, and which method do you use? I usually use a simple percolation of a non-solvent into a solvent to obtain good grade crystals, because we observed that at least for platinum, the simple evaporation of the solvent wasn't really effective. While the slow diffusion of a non-solvent was much more effective to obtain very good grade crystals for X-ray diffraction. If you fund it, yes, the funding for our research mainly comes from the University of Padova, okay, which every year fund our institute, our department, and then the different groups with some grants. Okay, so we have to apply with a project, and if you are funded, we have usually there are 2 year projects. But we also have some national funding system. There is what we call PRIN, which is the research of national relevance, which are more competitive, of course. But in the past, we used mainly for the bioinorganic part of my research. We obtained some funding from that side, so the national level.
Thank you, Professor Paolo.
Thank you, Ramachandran.
Okay, I think no more questions from audience. It was a great lecture on.
Yeah, as I said, I will send you.
Okay, you send presentation slide to my email. Then I will share with all the participants. Okay.
There they can find my email address, and so if anyone, any student, colleague wants to get in touch, I'll be available.
Okay, Professor. Okay, so now I come to the end of the sessions. It was a great and wonderful lecture on platinum chemistry at University of Padova. First of all, I would like to thank our research person, Dr. Paolo Sgarbossa, delivering a wonderful lecture with rich information. Thank you, Professor Paolo.
Thank you.
And also, I would like to thank Professor Mirto, editor of Inorganic Kimikaka, who joined us with a wonderful session. Thank you, Professor Mirto. I thank our management, director, principal, and head of the Department of Science and Humanities for their valuable suggestion to conduct this webinar. Then I thank all my dear colleagues, also extend their valuable support for the smooth conducting of this webinar. Last but not the least, I thank all the faculty members and students, participants from various university, colleges who joined in this webinar. Thank you to all of participants. Thank you, Professor.
Thank you, everyone. Thank you.
Thank you. So now the link is there in the chat box. All the PG students are asked to attend this test. Based on this score, they are called for our institute to hands-on training for the advanced instrument like infrared spectroscopy, UV visible, and fluorescence spectroscopy, and HPLC, and also CH instrument. So thank you, Professor. Thank you.
Thank you again.